Hello once again and welcome to the second session of our webinar series. Thank you for taking time out and being here today. For this session, I am pleased to say that Dr. Mahla Pudineh has accepted our invitation to talk about the next generation enabling technologies for diagnosing disease and monitoring therapy. As you know, this webinar is being held with a close collaboration between RASA and Omidafine and companies. Just to mention, we will have a live question and answer at the end of this webinar. So we have enabled the chat feature. You can find it on the right or left hand side of your screen, actually based on the language of your platform. So if you have any question, just write them in there, and if you missed anything, don't worry. We will be sending around the recorded version when it's available. Now I'd like to introduce today's presenter, Dr. Poudineh. Dr. Poudineh is an assistant professor and the director of Ideation Lab in the Department of Electrical and Computer Engineering at the University of Waterloo. She received her PhD degree in Electrical Engineering with a minor in Biomedical Engineering from the University of Toronto in 2016. Prior to joining the University of Waterloo, she completed postdoctoral training at the University of Toronto, Department of Pharmaceutical Sciences, and Stanford University School of Medicine in 2017 and 2019, respectively. She received her Bachelor of Science and Master of Science in Electrical Engineering, both from the University of Tehran in 2010 and 2012. Her research interests includes, but not limited to, developing biosensing approaches for therapeutics and diagnostics purposes and translating biomedical devices to the clinic. Moreover, she has plenty of valuable papers in prestigious journals, for example, in Nature Nanotechnology, Nature Biomedical Engineering, Small HCS Applied Materials, and Interfaces. Now, welcome, Dr. Poudineh. We are looking forward eagerly to hear from you.
Yeah, hello everyone. Can you hear me? You should be able to hear me.
Yes, of course. That's great.
Great. Thanks for the introduction, and thanks for having me as part of your webinar series. I'm very happy to be here. So I think I'll start by sharing my screen and presentation. Okay, once again, I'm happy to be part of this seminar series. Today, I'm going to talk to you about my work on next generation enabling technologies for diagnosis, disease, and monitoring therapies. And thanks for the introduction, but just a brief, you can say brief background about the history of my education and also research. And as it has been mentioned, I did my bachelor and master's at the University of Tehran in Iran, and after that I moved to Toronto, Canada to do PhD. And I did a postdoc at Stanford and University of Toronto, and as of January 2020, I started my own research lab at University of Waterloo, Department of Electrical and Computer Engineering. So during past 8 years of my research, I work on different projects for different applications. So I did some work at Stanford during my postdoc to develop real-time biosensors for continuous detection and measurement of insulin and glucose. And during my PhD, I worked at two platforms for cancer diagnosis and stem cell engineering. And now at my group at University of Waterloo, we are continuing to working on these projects. So my lab would have two main streams, one for the real-time sensing and the other for cancer diagnosis. So as my lab is like still new, I decided to share with you some of my works, from my PhD and postdoc, and at the end, I will talk about some future direction that we have started at Ideation Lab. So I'm going to start with the projects about cancer detection, and if we have time, I will talk about the real-time biosensing as well. So first of all, why early cancer diagnosis? So as all we know, cancer has been a leading cause of death in the society, and the thing that actually makes cancer a kind of serial killer in the world is not only the primary tumor. Basically, it's the spread of the cancer in the body of the patient, which makes it like dangerous. So, if we can diagnose cancer at earlier stages, the chance for successful treatment would highly increase. And this is something that has been known for a while now, for more than half a century. That if we want to diagnose cancer more effectively, we need to, if we want to, treat cancer more effectively, we need to diagnose it before the appearance of like any clinical symptoms. But to do this, first we need to know how the spread of the cancer or metastasis happens. This happens through the invasion of a kind of cancer cells that are called circulating tumor cells or CTCs. So during cancer progression, primary tumor cells start to shed these cancer cells into the bloodstream, and and these cells start their journey in the blood. So through the blood vessels, they can travel to distant organs, and then these cancer cells are finding a right place and all the conditions are met, they would exit the blood vessels, and in this location, the secondary tumor or the metastatic tumor would form. So this metastatic tumor would be another source to release more CTCs or circulating tumor cells in the bloodstream. And in this way, the cancer would spread into the body of patients. So if we want to detect cancer at earlier stages, we need to detect these circulating tumor cells at the very beginning. So, but the problem is that these CTCs or cancer cells are very rare. So there is less than, at the beginning, there is less than 100 CTCs per 10 to 9 blood cells. Like imagine like in a 1 ml blood, so it could be less than 10 CTCs. And we need to detect this low number of CTCs among these high number of blood cells. So we have been fortunate that we have engineers who are working with biologists and they're developing platform for a specific CTC isolation and capture. So in this slide, I'm showing some different devices that have been developed for CTC capture. So one of them is cellular, that is the only FDA-approved device for CTC capture and isolation. The other one is Vortex chip that enables CTC isolation through the based on acidification effect, or basically because the CTCs have a larger size compared to other blood cells, the Vortex chip enables its isolation based on these physical characteristics. So CTCI chip is another device that was introduced in 2013, and at that point it was a breakthrough. So it allows the isolation of the CTCs with the efficiency of like more than 99%. So the one technique that has been used for the CTC isolation is called immunomagnetic separation. So this technique is a well-known technique and has been used by both cell search and CTC-I chip. So in this technique, the cancer cells are specifically labeled with the magnetic nanoparticle through the antigen-antibody conjugation. So there are like some surface biomarkers on the surface of the CTCs which is called EpCAM, or epithelial cell adhesion molecule. And this EpCAM is only expressed, this only exists on the surface of the cancer cells, not on the surface of the blood cells. So when we are using like a capture antibody with like magnetic nanoparticles, like this conjugation can only be used for the labeling of the cancer cells, and the blood cells wouldn't get any label. So in this way, we are like basically making these cancer cells like magnetically labeled. And after that, the sample will be injected into a microfluidic device, like a channel, and an external magnetic force will be applied. When the cells are passing through this channel, the cells are getting captured if like the magnetic force can overcome the drag force. And in this way, the capturing of the cancer cells can occur. But the blood cells would be passing through the channel. So as I said, this is a very well-known technique, and it's called immunomagnetic isolation, and has been used by many technologies such as cell search and CTCIG. So these are so as I said, there have been many techniques developed for the CTC isolation. But the problem with these techniques is that they don't profile cancer cells. Basically, they are isolating a bulk of CTC population without doing any analysis on them. But as you can see in this slide, this is a tumor, and a tumor can be very heterogeneous. And a heterogeneous tumor would release heterogeneous population of CTCs. So some CTCs are benign and they won't do anything, so we don't need to be worried about them. But some of them are metastatic and they are invasive, and we need to detect these CTCs at earlier stages. In fact, it's known that during cancer progression, CTCs start to lose their EpCAM expression, and they became like low EpCAM. Basically, they would express low EpCAM on their surface. So the low EpCAM CTCs, they would have low number of magnetic nanoparticles on themselves, and they are more invasive. And we need to detect these low EpCAM CTCs at the earliest stage. So with this problem in mind, we decided to develop a platform that can differentiate between the non-metastatic CTCs and metastatic CTCs. Basically, we want to develop a platform that allows us to differentiate, that allows us to profile cancer cells. And this platform for sure has to be working with the blood cells, with the blood samples, because all these CTCs are in the blood, and should allow us to also profile low number of CTCs. So as I said, the CTCs are rare, so it could be less than 10 CTCs per millions of blood cells. During my PhD, I work on different devices for cancer cell profiling, and today I'm going to show you one of the main projects for CTC profiling that we developed a device, and we called it magnetic ranking cytometry, or MAGRC device. MicroC device uses immunomagnetic separation to isolate cancer cells, and it has 100 different zones for CTC profiling. Each zone of the device consists of two main features. One is an X structures that previously we showed that these X structures can adjust the drag force in the device, and also nickel micromagnet. So nickel is a ferromagnetic material, and it can be used to enhance the external magnetic field. The novelty of our work, it was that we increased the size of this nickel micromagnet along the length of the device in a way that close to the inlet when the blood is injected, the size of the nickel is the smallest, and close to the outlet of the device, the size of the nickel is the largest. And the platform, it was a sandwich between two arrays of the magnets. These external magnets are used to activate the internal nickel micromagnet. So we hypothesized that the high EPCAM cells, that they would have high number of magnetic nanoparticles on their surface. They would require like a smaller magnetic force for getting captured. So that's why they would get captured at the initial zone of the device, where the size of the nickel is the smallest. However, low EPCAM cells with the lower number of magnetic nanoparticles require a larger nickel micromagnet for their capture, and they would get captured at the final zone of the device, where the size of this nickel micromagnet is the largest. So at the beginning, we did the simulation and we tested this hypothesis. And we saw that cells with different number of magnetic loading or different EPCAM expression, they should be getting captured at different zone of the MagRC device. So this was interesting for us, and we moved forward and fabricated the device. For the fabrication, we use normal lithography and also soft lithography work to fabricate the MagRC device and the channels out of PDMS and also glass slides. As a first experiment, we challenged the MagRC device using the cancer cells with the known levels of EPCAM expression. So we spiked whole blood samples with the cancer cells, and we ran it through the device. And after that, we did some immunostaining. We basically colored the cells, and we count these cells manually. This is the result. So we saw that the cells, cancer cells, model cancer cell lines with different levels of EPCAM expression, they would get captured at different zone of the device. So this result was really interesting for us, and it shows us the capability of the MagRC device for sorting of the cancer cells based on their levels of EPCAM or their levels of invasiveness. So after this in vitro validation, we moved forward and we did, we characterized patient samples, like to characterize real CTCs. And for this, we had access to the patient cancers. So we had two cohorts of the samples, like patients with the localized tumors that their tumor didn't metastasize, and patient with a metastatic tumor. So in a patient with a metastatic tumor, we found that all the cells are getting captured at the final zone of the device, where the invasive CTCs are supposed to get captured. While for the localized tumors, we had the profile in all the zones of the MagRC. So this result showed us that the MagRC device can be used to differentiate between the metastatic tumor and localized tumor in the patient as well, and it can be used for early cancer diagnosis. So the next work that I would like to talk to you about it and show you some interesting ACM and microscopy images is stem cell engineering. So stem cells are a type of cells in our body that they can be differentiated to different cells. They can become different cells, like they would become nerve cells, blood cells, or muscle cells. And in fact, engineering these stem cells have been used for regenerative medicine a lot. So it has been shown that using micro and nano fabrications and without adding any chemicals, it's possible to regulate stem cells' fate, like what kind of cells they would become. In this project, we use a technique to investigate this. We had mesenchymal stem cells, and so there are type of stem cells. We cultured them on the gold nanostructures, and interestingly, we saw that without adding any chemicals, any growth factors, these stem cells they become neural cells. And so we use this, this is a fabrication process to make the lines of gold nanostructures. And this is a type of structures that we developed. So we put these structures in a chamber, and we use this chamber to culture the cells. So these are like some gold nanostructures that we use electrodeposition to making them, and as you can see, they could have very spiky structures. So this is our control when we culture stem cells in a petri dish in a flat surface, and we saw that that you can see like their morphology, how the morphology looks like. And this is like the morphology of the cells when we culture them on top of these structures. So as you can see, the morphology is completely different, and it shows that these cells, they really like the, they really like the gold nanostructures, and they become elongated along the length of these structures. When we observed this morphology, we saw that they look like neural cells. And that was interesting for us. So we moved forward and we tested the culture of the cancer stem cells on top of different structures. So we had structures with the low levels of spikeness, intermediate, and high levels of spikeness. So using like electrodeposition, you can create these structures. And we cultured the cells on top of these structures. So as you can see, as the levels of spikeness is increasing, the adhesion of the cells to the structures would increase in a way that like the cells starting getting only cultured on top of the structures with intermediate and high levels of spikeness. So in addition to observing the morphology and that the morphology looks like the stem cells, look like the neural cells, we did also immunostaining for the markers related to neural cells, like to touch one and map to their two neural biomarkers, and we saw that for the highly spiky structures and spikeness with the medium level, we saw that most of the cells are expressing these two biomarkers, so they become so it shows that they become neural cells, in fact. So here we showed that using the nanostructures, we are able to make neural cells without adding any chemicals. And this approach can be used later on in regenerative medicine for the application in regenerative medicine. So just wanted to know how much more time do I have? Like you can tell me. I just wanted to know if should I go to the next work. Okay, I think I might have like how much? Sorry.
I think we have enough time. For example, I think we have about 10 minutes for your presentation, and we can have questions after your presentation, and sometimes for speaking in Persian. But
sounds good. So we continue then to the next work. Like I guess in 10 minutes would be a good time.
Thank you.
Dr. Poudineh, excuse me. Excuse me, I think your voice has a technical problem, but now I think it's okay. Sorry,
Uh,
can you please say
sure, no worries.
something?
Can you hear me?
Yeah, of
Can you
course.
hear me?
That's okay. Now
Okay,
it's okay. Thanks.
okay, sure. Thanks. Okay. So the next work is the real-time biosensing. So it's a project that I did at Stanford, and we developed a platform for continuous measurements of the two important biomarkers, insulin and glucose. So continuous monitoring is important because we want to do like diagnose many diseases at earlier stages, and for this we need to know what is the levels of the biomarkers and how is their kinetics in the body. And it's important for the personalized medicine too. It's important that like for each individuals we will be able to monitor their own levels of the biomarkers. So, for example, one of the diseases that the continuous monitoring is really important, as all we know, is diabetes. So there are more than 400 million people worldwide are dealing with the diabetes. And over 50 million of these people require insulin injection. So in the development of the continuous monitoring continuous glucose monitoring devices or CGM devices actually change the whole world of diabetes. So these platforms allow the measurement of the glucose in a continuous way. So the levels of the blood glucose can be tracked in real time using like CGM devices. So for people with the insulin injection, recently APD or artificial pancreas devices have been developed. In these APD devices, the level of the glucose is measured using CGM, and then based on this glucose level, insulin will be injected to the patient using like an insulin pump. So this is people with diabetes started using this APD system, but there is a big problem with these devices. Is that the level of the insulin is not measured directly, and the insulin injection is done based on the level of the glucose in the blood of the patient. So if like and this could make like that this could actually inject an like excess of insulin to the patient, because we're not measuring the level of insulin directly, and this excess of insulin could lead to a situation it's called hypoglycemia, when the level of the blood glucose it would come very low, and actually it would have like many other complications that the patient would go on into coma and many other problems. So it's needed then, in addition to the glucose, the insulin to be measured, and the insulin to be injected based on the levels of the levels of the insulin in the patient. So we need to monitor both insulin and glucose in real time. But continuous monitoring of the insulin is challenging, and it's mainly because that it's really low. Its concentration in the blood is extremely low. So for the glucose, the levels is in the millimolar, and for the insulin, this is in the picomolar. So you can imagine this is around 9 order of magnitudes difference between the glucose and insulin. And we need to do this measurement directly in whole blood. So that's why there isn't any current technology that can measure like insulin in addition to the glucose in real time. And that was an obstacle in the field. So based on this motivation, we started to think of how we can develop a platform to measure the kinetics of the insulin in vivo and in real time. So we use started to think of like that we need to miniaturize like molecular diagnostic into a small microfluidic device. One of the important molecular diagnostic approaches is called ELISA, that has been used in the lab for the measurements of the proteins and biomarkers in the patient. But for ELISA, it's like the blood drawing from the patient has to be done, like for example, if you want to get a kinetics, it has to be done every 10 minutes. And we can't get a real-time measurements, basically. So we decided to miniaturize ELISA and make the ELISA a real-time technique. And that's why we called our platform real-time ELISA. So in the real-time ELISA, there are three main modules, like a mixing module, a depletion module, and a detection module. Inside the mixing module, the blood samples is mixed with our reagents. So we are detecting the insulin and glucose using a bead-based assay, where the insulin they are getting captured on the beads and they are fluorescently labeled. So our reagents are the beads with the captured agents on them, and the blood samples would contain the target. So inside this mixing module, this capture can happen. After that, we need to be able to see these beads, fluorescently labeled beads. We need to isolate the beads from the rest of the blood cells. Basically, we need to deplete all the blood cells. So we use a technique for separating and sorting of the bead based on their size. So we use 15 micron beads. So this 15 is larger than the majority of the blood cells. So the blood cells would go to the base while the beads are going to our detection channel. And inside the detection channel, as the beads passing through, we would be able to measure the fluorescent signal intensity. This is a whole like an overview of the RT-ELISA device, but we used our bead assay is, as I said, we are using like a bead-based assay that we have like the capture agents on the beads, and upon the binding of the target, these beads would fluoresce. This is like our results for the glucose measurements, and as you can see, as we are increasing the concentration of the glucose, the fluorescent signal intensity would increase. And this is the results for the insulin detection, that we use a capture agent or capture antibody on the beads, and we had detection antibody with a fluorophore. And a sandwich assay would form on top on the beads. So here again, as you can see, as we are increasing the concentration of the insulin, more target would get captured on the beads, and the fluorescent signal would increase. So inside the mixing module, this mixing would happen, and basically it's an incubation time. So if we are not using microfluidics, this incubation would require like several hours to be completed. But the beauty of microfluidic is that like the reaction can happen faster, and this would act like the microfluidic rate the reaction. So using like this mixing module, we were able to reduce the time of the incubation to less than 1 minute. And actually, this is like this allows us to make like RT-ELISA a real-time platform, because we don't need to wait several hours of incubation. And we had a depletion module, which is really important for the separating of the blood cells from the like the beads. So as you can see before separation, so here like the beads are getting lost in the whole background of the blood cells. But after the separation, we are able to see the beads. So this is how the platform looked like, the microfluidic device with the mixing, depletion, and detection modules. And this is like the platform in a fluorescence microscope. We also built on our own fluorescence microscopy. Like we make a mobile and more like a mobile and miniaturized microscope settings that we can move it to for our like future animal experiments into the animal facility that we were doing that. Here is also just a video to show you how the beads are passing. So we use a spatial multiplexing that on top of the channel here, the glucose beads would shine, and on the bottom of the channel, the insulin beads would shine. So as the beads are passing, we would measure the fluorescence signal intensity, and this intensity is correlated to the concentration of the glucose and insulin. So some in vitro results that we spike different concentration of the insulin and glucose into whole blood, and we ran it through the device, and we use our calibration curves to find out the concentration that the device is measuring. So, for example, we spiked 15 millimolar glucose and 1.6 nanomolar of the insulin at the same time, and we did measurements. After that, we washed the device and spiked other concentration and measured them. After this in vitro testing, we moved forward and we did some in vivo monitoring. So in this in vivo monitoring or monitoring in real animal, we connected the RT ELISA through a peristaltic pump to a rat. So the peristaltic pump is a pump that can at the same time withdraw the blood and also inject it to the RT ELISA. And we injected our our reagents that are the beads that are capturing insulin and beads, and we did continuously measure the level of insulin and glucose in a rat. So there's some data that we got. So each of the blue dots is showing the RT ELISA measurements, and the orange square is showing the measurements using like our standard technique. So we collected blood every 5 minutes, also from the tail of the rat, and for the insulin we did ELISA to measure the insulin level, and for the glucose we use a glucose measure to measure that, and we saw a very good correlation between the RT ELISA and the continuous measuring data. So these results show actually the first real-time measurements of the insulin that can unravel its kinetics. And we also did some experiments that we injected the rat with different type of insulin. So there are different type of insulin that can act short or faster. And so we injected into the rat, and we saw different kinetics for these two different type of insulin. So in summary, RT ELISA can simultaneously measure glucose and insulin in vivo in animals. And it's so another thing that I wanted to mention about this platform, that it's a general and universal platform, because we are using antibodies, and easily we can just change it to other affinity agents for other biomarkers, and can be used for the measuring of the kinetics of other biomarkers that are interested, for example, other disease-related agents. So this is end of my presentation, and for the work that I have done at University of Toronto and Stanford University during my PhD and postdoc, and I'm very privileged to have great mentors during my research time. So now at University of Waterloo, I have started my own lab, and we are working on two main projects. One on cancer diagnosis that we are looking into measuring other cancer markers, like such as exosomes and circulating tumor DNA that's similar to the CTCs. There are also biomarkers that are found in blood. So we are developing devices to extract and characterize them. And also in another direction in the lab, we are working on the real-time and continuous measurements. That our goal is to make the RT ELISA platform a miniaturized platform, like that can be used as a wearable device for the continuous measurements of different like biomarkers. Thanks for listening to my talk, and I'm happy to take any questions.